In the previous lecture, we have already seen the part of a WBC, that is leukocytes, that is called as granulocytes. Now in this lecture we are going to see agranulocytes and third part, that is called as platelets. Okay, so talking about agranulocytes, we know that the cytoplasm of a granulocyte is without granules. Okay, granules are absent. That's why they are called as agranulocytes. Okay, they are about 28% of the total W bases. Okay, W bases in that granulocytes and agranulocytes. Granulocytes are 72%. So remaining 28% is agranulocytes. Okay, that agranulocytes are formed from the lymphoid stem cells. Okay, and they divide by mitosis. Now it is a characteristic feature of agranulocytes. They can divide by mitosis. Okay, agranulocytes, their nuclei are larger in size than that of the granulocytes. Okay, nucleus of agranulocytes are larger in size but not lobulated like that of the granulocytes. As in the granulocyte it is lobulated, 2, 3 up to 7 lobules are there, but in the agranulocytes there are no lobules are present in the agranulocytes. The agranulocytes are mainly divided into two types. These are nothing but lymphocytes and monocytes. Okay, you can see the diagram of lymphocytes and monocytes on your screen. You can easily identify lymphocytes and monocytes. Okay, so both one by one we are going to see. Lymphocytes are smallest of all W bases and monocytes are largest of all W bases. So this is a major difference between the lymphocytes and monocytes. Lymphocytes are smallest of all W bases and monocytes are largest of all W bases. So remember student that in total WBC, smallest and largest W bases are present in the agranulocytes. Lymphocytes are smallest and monocytes are largest of all W bases. Okay, the lymphocytes are having large spherical nucleus. Okay, so the nucleus of lymphocytes are spherical in shape. They are large but they are spherical in the shape. Here in the monocytes the nucleus is large but they are kidney shaped. So there is a greater difference between the lymphocytes and monocytes is that their nucleus is different, structure of nucleus is different. In the monocytes it is about kidney shaped and in the lymphocytes they are spherical in the shape. The lymphocytes constitute about 25 to 30% of total W base and the monocytes are about 3 to 5% of the total W bases. This is a third difference between them. Okay, then in the monocytes, okay, the monocytes are give rise to the macrophages. Okay, that macrophages are mainly phagocytic. That means monocytes are phagocytic. They give rise to macrophages which are phagocytic. That means monocytes are phagocytic. Phagocytic means what they functions, what type of functions they done. They destroy the damaged tissue and bacteria by phagocytosis. That means monocytes are useful for to destroy the damaged tissue and the bacteria by the phagocytosis. So phagocytosis is the main function of the monocytes. Okay, this is about the monocyte. Now lymphocytes are again divided into two types. Okay, that is B lymphocytes and T lymphocytes. Now, why it is called as B lymphocytes and T lymphocytes? Okay, uh, first we will see the B lymphocytes. Okay, B lymphocyte, it is mature in the bone marrow. Okay, now it is remember student that B lymphocytes and T lymphocytes both are originated in the bone marrow. Okay, originated that means they are evolved from the bone marrow, but B lymphocytes are mature in the bone marrow. Okay, they originate from the bone marrow and they are mature in the bone marrow. That's why they are called as B lymphocyte. B is for bone marrow. Okay, so that's why these are called as B lymphocyte. The B lymphocytes are responsible for the humoral immunity, or we can simply call it as antibody production. That is very much important function in our body. Antibodies. Now you can understand that in this pandemic situation, COVID-19 pandemic situation, the person's antibodies becomes very poor. Okay, that's why their immunity becomes poor. So the production of antibody is the function of B lymphocyte, and B lymphocyte, it is a part of a granulocytes. A granulocyte is a part of WBC and WBC is a part of blood. So blood count or all the components of blood, if it is correct, we are healthy. If one of the part of the blood or fragment of the blood it is missing or it is less, then it is directly affects to our immunity system. So B lymphocytes are responsible for humoral immunity or simply antibody production, and these B lymphocytes are highly specific to the antigen. That means a particular antibody only destroy the particular antigen and not others. Okay, for each antigen the antibody is different. Okay, so they are specific, highly specific to each and every antigen. Then talking about the T lymphocyte. Okay, that T lymphocytes are mature in the thymus. T for thymus. Now they are originated from bone marrow, but they are mature in the thymus. That's why these are called as T lymphocyte. These T lymphocytes are responsible for cell mediated immunity. Now this is also immunity, but it is cell mediated immunity. That's why this is the difference between B lymphocyte and T lymphocyte. B lymphocytes are humoral immunity and T lymphocyte are cell mediated immunity. Again, in the T lymphocytes there are T cells are present. There are mainly 4 types of T cells are present. These are namely T cells, killer T cells, memory T cells, and suppressor T cells. These are types of T lymphocytes. Okay, and they are protective in function. Okay, so this is about the lymphocytes. This is about the types of a granulocyte, that is lymphocyte and monocyte. Lymphocytes are smallest of all WBCs. Monocytes are largest of all WBCs. About 25 to 30% of total WBC is lymphocyte and 3 to 5% is monocyte. Okay, lymphocytes are having again 2 types, B lymphocyte and T lymphocyte. B lymphocyte mature in bone marrow, T lymphocyte mature in the thymus. B lymphocyte responsible for antibody production and T lymphocyte responsible for the cell mediated immunity. Monocytes, they give rise to the macrophages. Their main function is to phagocytosis. Okay, then coming about the third coming to the third element of the blood, that is called as platelets, or we can also call as thrombocytes. Okay, thrombocytes or platelets are small oval shaped shell fragments without nucleus. Again, here also nucleus is absent, but they are oval shaped cell fragments. Now, fragments of which cells? The fragments of cellular fragments of megakaryocytes. Okay, platelets are nothing but the small oval shaped cellular fragments of megakaryocytes without nucleus. Okay, the normal count of platelets in the blood is about 2.5 to 4.5 lakh per mm cube of the blood. Okay, you can see the normal count of platelets in the blood reports if you are having. Okay, all these components we are studying, it is in the blood reports. Okay, so you can see the normal count, how it is calculated, and what are the components of blood are present, and how the tests are there, you can see. Okay, so normal count of platelets is about 2.5 to 4.5 lakh for mm cube of the blood. If there is a decrease in the number of platelets, in such condition it is called as thrombocytopenia. And this thrombocytopenia or decrease in the number of platelets will causes internal bleeding, that is also called as hemorrhage. Now it is very much important. Now platelets are very important, because if there is a decrease in the number of platelets, then there will be internal bleeding or hemorrhage in the body takes place. Okay, now these thrombocytes, what they functions? Thrombocytes secrete factors responsible for the blood clotting. Now after completion of this point, we are going to study the blood clotting. In that, whatever the factors are responsible, some of the factors are secreted by these thrombocytes. Now again, thrombocytes are important for the blood clotting. So they secrete a vasoconstriction called as serotonin. They secrete serotonin also, and other factors, blood clotting factors, they are secreting. So this is about the platelets. Okay, now coming to the most important part about the blood, it is nothing but we have seen the three of the component of blood, that is RBC, WBC and platelets. Red blood corpuscles, white blood corpuscles and platelets. Now let's talk about the blood clotting or coagulation of blood. Remember student that it can be asked in your exams. Define blood clotting, or what is blood clotting. The definition is very simple. It is blood clotting is a process of converting liquid blood in solid form. Okay, so liquid blood is converted into solid form. It is called as blood clotting. We know that if there is any injury to our body, after some period, the blood is going to become a dry. That is called as a clotting of blood. Now, how this blood is going to be clot or it becomes dry? This process we are going to study. This process of the blood clotting is initiated by intrinsic or extrinsic pathway. So you can see one chart on your screen. Okay, this chart starts from the extrinsic and intrinsic pathway. So there are two pathways for the blood clotting mechanism. Extrinsic pathway is what? Extrinsic pathway starts when damage to the tissue outside the vessel. Okay, so damage to the body outside the vessel, it is called as extrinsic pathway. Now what is intrinsic pathway? Damage to the blood vessel, it is called as intrinsic pathway. So by both intrinsic and extrinsic pathway, the blood clotting is done. Okay, so talking about the extrinsic pathway, in extrinsic pathway, damage to the tissue outside the vessel, then the thromboplastin present in the tissue, okay, that is going to be function. And in case of damaged blood vessel, the cascade of clotting factors, there are about 12 clotting factors are there, okay, namely 1 to 12, Roman 1 to 12 clotting factors are there. So in the extrinsic pathway, tissue thromboplastin is function, and in the intrinsic pathway, cascade of 12 clotting factors are functioning there. They both functions what? They activates the factor 10. Okay, inactive factor 10 into activated factor 10. You can see in the chart. Tissue thromboplastin and cascade of clotting factors both can activate factor 10. Okay, what is that factor 10? Prothrombinase. Okay, prothrombinase is factor 10, and thromboplastin and 12 factors activate inactive prothrombinase into the active prothrombinase, and this enzyme prothrombinase inactivates the heparin. Heparin is the enzyme, inactivates the heparin and convert inactive prothrombin into active thrombin. Okay, you can see activated factor X, what it performs. Activated factor X converts prothrombin into thrombin. Okay, so thrombin is formed here. Now this thrombin, what it functions? This thrombin converts soluble fibrinogen into insoluble fibrin. You can see in the diagram, thrombin from prothrombin, thrombin is formed, and this thrombin, what functions? What it functions? It converts fibrinogen into the fibrin. Okay, and this fibrin forms a mesh in which platelets and other blood cells are trapped to form a clot. Now they make a mesh. The fibrin forms a mesh on the injury part, and in this mesh the platelets and other blood cells are going to trap to form a clot. So this is about the blood clotting or coagulation of blood. It is very simple mechanism. In both way, extrinsic or intrinsic pathway, they both coming to the same manner. In extrinsic pathway, injury outside the blood vessels are called as extrinsic pathway, and damage to the blood vessel, it is called as intrinsic pathway. If there is a damage to the blood vessel, then 12 clotting factors are chain of 12 clotting factors, that is called as cascade, it is going to be activated. And if it is into the outside the vessel, then tissue thromboplastin is activated. Both tissue thromboplastin or cascade of clotting factors are going to activate the factor number 10, that is prothrombinase. Now that prothrombinase converts prothrombin into the thrombin. Again this thrombin is going to convert fibrinogen into the fibrin, and finally this fibrin, it makes a mesh like area, and in that mesh like area other components such like that of the platelets and blood cells are going to be trapped. This is called as a formation of clot. Okay, very simple mechanism is there. That's how the blood cells are completed. Okay, in the next lecture we are going to study about the heart. Okay, its external internal structure is working mechanism. Okay, we are going to see in the next lecture. Okay, thank you.